Jo. Na poloukoli, že to nahráváš. Nahráváš tady.
Já to nahraju sobě a pak to pustím. Já nechci, aby se to nahrávalo třeba. Jo? Okay, so welcome everyone. So here we have Alice Bucek who arrived to our department. So he's starting to be here around 6 tomorrow. He will be using with us office. Since tomorrow, sorry. Anyway, he will be sitting tomorrow. You can find him in the office of Vojta until we move, and then he will be moving with us to the new building. And Alice will be the PI of the new laboratory of symbiosis.
Insect symbiosis. All the fancy names were already taken, so I had to navigate to the blue.
Yeah, so he's going to introduce himself today, a little bit his work. So enjoy. And this is also the last seminar of this season, so we will meet each other in September again. Okay, thank you.
Thanks, Dr. for the kind introduction. And yeah, hello everybody. I literally moved to České Budějovice 3 days ago. So I've never worked here, visited České Budějovice just a couple of times. I'm from Moravia, from the eastern part of Czech Republic. Then I did my university studies and PhD in Prague, but the last 6 years I've been in Japan. And now I got this Gatcher Junior Star grant for 5 years, which is actually running from this January for the next 5 years. And I complicated my life a little bit. I applied for the grant with the university in Prague. Now I'm transferring it to here. It was not yet approved, but we were told it will be approved. So I'm already committing, I'm moving in, and 22nd June, hopefully everything will be settled administratively. So I might have a bit too ambitious plan for today to explain like how I went from PhD via postdoc to what I will be doing here with you. I will see how is the time. I might be then skipping something, so don't worry if I will skip something, and we can always chat about it later on. So I defended in 2016. My background is biochemistry. At Charles University at the chemistry department, but basically my whole life I spent, I mean my whole PhD and master and bachelor I spent at the Institute of Organic Chemistry and Biochemistry in Prague. Just like the first month after I arrived to Prague and started university, I sent email to Dr. Iva Pichová there if I can join her lab, and I've been there until 2016. So it was a very nice environment. Then 2017, I moved to Japan and had there pretty long postdoc in Okinawa. It's place which is hard to leave, like a subtropical nice island with quite cutting-edge research institute with a lot of money. Japanese government is pouring a lot of money into this. It's also as a sort of not subsidy, but a plan to support Okinawa, because Okinawa prefecture is one of the least developed prefectures. So the Japanese government have a couple of plans how to stimulate development there. So one of them was, okay, let's build this institute, which they claim has ambitious ambition to produce Nobel laureate, which I don't know. But anyways, they managed to. The research was not really done there, but they managed to recruit a couple of years. Maybe you heard about Svante Pääbo. So that's actually a guy who got a Nobel Prize. So the research he got a Nobel Prize for on ancient DNA and evolution of Homo. He mostly did not do in the institute, but they managed to get him, and now he has a lab there. So they are sort of maybe on the track. And now. Last November, I learned that I got this 5-year research grant from the Czech grant agency. So I was very happy to decide to move back to my home country. So what I've been doing, I will be skipping through this, I think, quite fast. So don't worry if you can't follow all the small details. At the Institute of Organic Chemistry and Biochemistry, I've been mostly doing chemical ecology on a couple of insect models on Manduca sexta. My work would be basically try to find out which genes and enzymes are involved in biosynthesis of pheromones. In case of Manduca sexta, the sex pheromones. Then later on, as the next generation sequencing methods, and that was really the time I started when next generation sequencing methods were not really existing, or they were sort of emerging. So once it became available, I started to use also transcriptome sequencing to find genes which are expressed in the pheromone glands, and then I would be taking these genes, putting them into yeasts to see what these genes or the enzymes they encode, what they are producing, if they produce the pheromones. I would be mutating the genes to find out how much we have to mutate a gene which is producing pheromones, so it starts to produce different pheromones. So all this was about learning what are the mechanisms of evolution of this pheromone language, and that was done on one species, this and then I played also with other model system, bumblebees, where we were looking at another enzyme. So yeah, this one enzyme is introducing double bonds, like one particular step, but the position of the double bonds in the pheromone, that's determining the species of the pheromone. So that's like that's the information containing part of the molecule. Then I also work with bumblebees, which produce similar pheromones, but they have alcohol groups. So I was looking at enzymes which produce this alcohol group from carboxylic group. And we found that in the, like one of the interesting things we found is that in the common ancestor of bombus and or bombine and stingless bees, one gene family expanded, which probably then involved what kind of pheromones are bumblebees using, and also what kind of pheromones are stingless bees using. So that's what I've been playing with during my PhD, and a little bit, and that's what I thought, and that's how I got to termites. So soon I will start talking a bit more about termites. I met friends, mostly I was spending time with them in pub, but these crazy dermatologists in the pub, they still keep talking about termites, and so they got hooked me. And I thought, okay, that might be something I will continue on postdoc, because I was already with the other projects very much interested in the biosynthetic capabilities of insects, and termites are also very cool in this sense, especially their soldier dust, which can produce a lot of quite crazy compounds, like for example this nitrated compound, which we still have no idea how it is produced in this living organism. That's I believe the only kind of this compound anywhere as a secondary metabolite anywhere in the animal kingdom. And another example, also as a biosynthesis of defensive compound, these hydroquinones, and then their conversion to non-toxic hydroquinones, and then their conversion to benzokinones. So, but in the end, I did not go this way. I just contributed more as a technician, isolating RNAs, processing samples, and so on. And but importantly, I met then my future postdoc PI, Thomas Bourguignon, who in 2016 got his position, his PI position in Japan, and he offered me a postdoc. So that's how I got to Okinawa. That's here in the middle of nowhere, fairly far from the rest of Japan. It's like 2.5-hour flight to Tokyo. This is how it looks like. Really, really nice place. The institute is somewhere already in the part of the island where it's mostly covered by jungles. I would say like southern third of the island is heavily urbanized, almost like continuous urbanization. But in the northern half, that's like continuous jungle, just some small towns on the coast. So, and the institute is somewhere in between. So it was mostly 6 years of village life, and there's just a couple of very fancy hotel resorts. But it was quite a peaceful life with a nice coral reef all around. This is Thomas. So that's how I got to the postdoc, and now I will tell you a little bit what I've been doing during my postdoc, and then how this directed me towards what I will be doing here. So I might not need to introduce again termites in terms of how abundant and ecologically successful they are. If you look at some of the estimates of the biomass, they represent up to 25% of the terrestrial arthropod biomass. So they are extremely successful, not really here, for example, in Czech Republic. They are mostly distributed in tropics, to smaller extent in subtropics. They are not surviving very well winters. And it's not only, but again, I probably don't have to stress this. For different audience, I think I changed title. Usually here is not only pests, because mostly people recognize termites as pests. So there is many more services, not only eating our wooden structures or eating our crops they provide. For example, in an experiment which was run in Borneo, in two kinds of plots, one of them with experimentally removed termites, one of them were untreated, where termites remained. There were a couple of significant and large changes. For example, the leaf litter decomposition after eradication of termites decreased dramatically, and also, for example, survival of seedlings decreased dramatically. And this was most significant, and the effect was largest during drought periods. Or similar experiment in Australia. In this case, they eradicated at the treatment plot both termites and ants, and then had a control plot without any treatment, and that was on the fields, and they looked at the yield of grain, and they could see where these two groups of social insects were eradicated. The yield, which is here, dropped really dramatically. So yeah, termites do a lot of useful stuff in our ecosystems, and I mentioned they are extremely ecologically successful. There is probably a couple of life traits which allow them to reach this success. It's their eusocial organization, ability to build large colonies, well defended. Related to the defense, the primary defense is their termites' nest, but then they have also dedicated defensive caste, which I will talk a bit more about in a second. And then nutrition. And that means their ability to digest and utilize wood or soil. That means substrates which are plentiful, but not that many animals can really utilize that. I've never really researched much into usability, but I did some work on trying to understand evolution of these two life traits. So that's what I will focus on. And I've been typically doing it by building phylogenies. So I will give you this schematic overview, just so we know how the termite diversity look like, and then I will talk a bit more about particular groups of termites. So termites are usually the cockroaches. Their common ancestor with Cryptocercus cockroach, and or the common ancestor of Cryptocercus cockroach and all termites is some 150 to 200 million years ago, dates back to this period. Then we have this paraphyletic group of lower termites, which is some 20% of termite diversity. And then some 50 million years ago, something happened. I will mention what we think might have happened. And family termitid emerged, which is now 80% of termite diversity, and also takes disproportionate share of the termite abundance. Here I'm showing you, these are soldiers. The workers are rather boring, at least morphology-wise, while soldier have all these very interesting mandibular adaptations. So that's something, let's say, which was known before I entered the field. Then my starting project was, until I started my postdoc, the phylogenies first were built on like one PCR amplified loci, then a couple of PCR amplified loci. Then Thomas Bourguignon IPI did incredible amount of work using mitochondrial genomes at the scale which was not done before, with like first tens, then hundreds of species, and with this access to the globally representative termite collection, it was able to really refine at that point our understanding of termite phylogeny. My project was to have a slightly smaller sampling, but use transcriptomes. So at that time, that was the first phylogeny where we used, where for termites, large number of genetic loci was used. That's I'm not sure how familiar you are with molecular phylogenetic methods, but there is, especially in branches which diversify rapidly, it's quite important to have a larger number of genetic loci, because having phylogeny based just on one or few genetic loci might not allow you to really resolve that that branch or that part of the termite tree of life. So, and then this has follow-up. Colleague of mine is now built ultra-conserved element-based data set. Ultra-conserved elements are also genetic loci, but while in transcriptomes, these are genes which are expressed, these ultra-conserved elements are like small portions, which are always in a very similar, with a very similar sequence or almost identical present across all the animals you want to reconstruct your phylogeny for. So, for example, here for termites, and that has many advantages, because for building phylogenies with transcriptomes, you need rather intact RNA, and RNA is inherently not very stable, while DNA is pretty stable. And also, ultra-conserved elements are rather short, so even you can use, for example, dry using specimens. So, or for example, for this work, quite some type specimens, some of them older than 100 years, and stored in like very poor conditions, were used. So that's quite promising. But what we learned from the first, let's say, scale phylogeny, we refined position. So importantly, we learned that the phylogeny is based on mitochondrial genomes were actually quite correct. So that's not a very fancy conclusion, but we were not sure until that point. So that was one important conclusion, but it's hard to publish as well. So we then in the paper we published, we focused on one particular branch of the termite tree of life, where we actually found a very interesting incongruence, disagreement with all the previous phylogenies. So that's here at the around the Termitidae family, and in green are fungus-growing termites. These are termites which we think might cultivate bacteria. Not much is known about them. It's a side project I was working on, but did not put much effort into it. So this is still very much in progress. And for a minute, Termitidae, it's a soil feeding termite group. And it was not really clear how these subfamilies are related and why it matters. Because in the evolution, I mentioned that something happened when the Termitidae family emerged. That means that the family which encompasses some 80% of termite diversity and most of their abundance. The thing that happened was a big switch in the symbionts they have in their guts. All the other termites have mostly bacteria, which are responsible for the digestion, but the other termites are mostly protists, which are responsible for the digestion. But then Termitidae, this most successful termite family, lost the protists and functionally replaced them with bacteria. And some of these phylogenies were suggesting that the termite subfamilies, which let's say externalize digestion, that means they, for example, Macrotermitinae, they have some fungal gardens which they use for digestion of wood, and Spherotermitinae, they have some hypothetical bacterial garden they use for the digestion of wood. This was stimulating hypotheses, which were the most parsimonious explanation of these trees, that externalization of digestion was some intermediate step, how the lower termites lost then protists, and then later on were replaced by bacteria. So, but so the important or the interesting thing, the result of these transcriptome-based phylogenes is that actually these two groups are sister. So then means that the externalization might be just a direct trait, which is shared by this fungus growing and this potentially bacteria growing termite. So that suggests that the switch in the symbiosis in termites might not have to occur via externalization of their digestion. I think I will skip over this. I've been also involved in looking into some other symbionts or parasites of termites. In the transcriptomes I generated, collaborators found quite a diversity of termite viruses, and yeah, 2020 was not a perhaps great year, but a very good year to publish things about viruses. So this one, despite being very descriptive, it's being and in MTPI journal, it's being still quite well cited. And I've also collaborated quite a bit with our PhD students on looking more into detail at the gut bacteria. And so, for example, here, these are from gut bacterial metagenomes inferred enzymatic families with different activities. And this is a termite tree of life. So that was a rather big project with some 200 samples across the whole termite tree of life, and we were looking at what the bacteria can, or what's the, let's say, potential of the bacteria inferred from what they have in their genomes, what they can do, what they can't do, and how it relates to the ecology of the termites, or how this evolved through the evolution of termites. I will not go into detail. Then I've been involved in more focused work on particular termite subfamilies. Mostly we were using mitochondrial genomes, build the phylogeny, and then often we would have typically nice sampling, nice global sampling, and then we would be inferring time-calibrated phylogenies using some publicly available fossil evidence, and then also inferring historical biogeographies, how the termites colonize the world. So that's an example of one lower termite group. Here's another focused paper on another group. Different kind of focus. This paper was somehow different. We worked on. I don't have. So these are papers from Menglin Wang, our PhD student. This one as well. It's the most recent. I don't have the reference. So this was a bit different strategy. We had quite a sampling of termites from Madagascar, so we were able to infer how many times termites colonized Madagascar, and what was the time frame of the colonization. Again, I will not go into details. Okay, I think I might skip a few things, so I get to what I will be actually doing here with you, although all these methods I will be to some extent also using. That's it's the second largest after termite, the second largest termite family. I think I'll skip through this one and continue with. I was telling what everything we could conclude from the phylogenies, but one also take-home message from the transcriptome-based phylogeny was that we did not resolve everything, despite having like thousands of genes. There is still this, let's say, wall of short branches, which encompasses. So this is Termitidae, and still most of the Termitidae species, which is like the most termite species in the world, and they're the largest share also of their abundance. That's not really clear how they relate to each other. The problem is that this group diversified very rapidly. Some, let's say, 25 to 40 million years ago, but a lot of these branches are as short as 1 or few million years. So this is extremely hard to resolve, and that interferes with our ability to understand a lot of things which happen here. How some of the interesting life traits of this large termite evolved. And one of the interesting life traits, and I will very briefly show how I try to infer how some life traits evolve despite not having fully resolved phylogeny. So an example of a life trait I'm still working on quite a lot. This work was not yet published, although I'm now finishing the manuscript. That's the evolution of the termite mandibles. That's a quite exciting thing. There you have there are a lot of variation, like huge mandibles or combination of this chemical gun in the form of nasus, which is connected to a large reservoir of chemical defensive substance, or in some soldiers, the mandibles are completely lost or are like extremely rudimentary and non-functional, or some exciting things like polymorphic, here dimorphic soldiers, one of them biting, one of them with this small chemical gun, or like, for example, phrymosis. But I think the most exciting is this very weird thing. It almost looks like malformed, but these mandibles are actually adapted for not biting, but for so-called snapping. Snapping means, and yeah, this means snapping. So normally it would be biting, but snapping, it's a very different strategy where the mandibles press against each other and they elastically deform, and at some point they slip, and always the left mandible is then accelerated and is leading the strike. So that's quite peculiar strategy, or especially because it evolved only maybe once or twice in ants, and it never evolved as far as we know in other insect groups. So that seems to be quite weird, but maybe the explanation is that once you have such mandibles, they are not compatible with feeding anymore. Like these termite soldiers are fully dependent on the workers to mouth feed them. So you probably need social insects to be able to afford this craziness, and it's extremely fast. So this video we looked at, if I can play it once more, and but there will be some more. Yeah, this video was like 100 of enabling. So it's basically a spring-loaded mechanism. And here we might have some more. Here is the effect on an opponent. So it's basically tears away whatever is in the path of the mandible. Here, please pay attention here. It's Solenopsis and one snapping termite species. So that's the stereotypical movement. Basically, the termite lobs the mandibles, then approaches the opponent, and then the mandibles are a little bit more, and then it slips and hits the opponent. And it's very much like a finger snapping action. So even somebody published a paper about the biomechanics of finger snapping. So your finger snap is like the peak speed of your tip of finger is some 10 meters per second. But termites can do some 70 meters per second, which is already quite a bit faster than the, for example, maximum speed on the highway. Then, and this is like succession how the papers were published. Some ants can do even faster, but it's still very analogous, although independently evolved thing. And then the latest application was about termite reaching with the tip of the mandible reaching almost 500 kilometers per hour. And so we were wondering what's actually where it will stop. We knew that snapping is evolved likely many times independently in termites. So we basically wanted to look at every independent origin of snapping, measure the speeds to understand if maybe there is some biological limit for this speed, and or if termites always evolve very similar snapping mechanism, or if there are some unique, let's say, design solutions. So to do that, yeah, here is just illustrated that in red, mapped onto the transcriptome-based phylogeny, there were for a couple of independent origins. Yeah, here is also one. And snapping in termites is actually quite prevalent. It's in some 40 genera, some 300 species, which is like 10% of termite diversity. So despite being like very weird strategy, it's quite prevalent. And yeah, just an example of what I mean by convergent evolution. There are some more famous examples. So this seems to be another quite remarkable example of, let's say, rather complex biomechanical adaptation, which evolved multiple times independently. So perhaps similar to echolocation or powered flight. So here are examples of how the termites actually look like on the termite tree. The mandibles can be symmetrical, or what I've also already shown you, this very weird asymmetrical. So in the symmetrical, I think I explained that correctly. In the symmetrical, both mandibles would be striking. So then the termites can, like, for example, turn to the opponent and hit it with either mandible. In the asymmetrical, only the left mandible is striking. The right mandible is usually rather short and just like loading the left mandible. Then there are even species, termites with dimorphic soldiers, where both mostly symmetric and very asymmetric is present. And snapping evolved in termites across their scales. So from some of the largest termite soldiers, these are relative volumes of the head capsule, to some of the smallest termite soldiers. So it seems to be strategic efficient across the scales of termites. So here are some more videos. And we did not record them only because they look quite amazing. Here, by the way, you can see some liquid. That's defensive secretion, which is being dispersed. So that's also something we did not know before we started IB. Here, I mean, who did all these recordings. I was not really involved in the recording of these things. So yeah, the dispensing of the secretion is something which was not yet published. And we recorded these so we could quantify the energies and then look more into detail how the different strategies compare in terms of the performance. And to be able to calculate, we wanted to calculate the performance in terms of how much the termites, let's say tissue-wise, invest into it. And we thought it would be good to, for example, quantify the volume of the mandibular muscles. For that, we used X-ray microtomography, and here are already the segmented images. I will have here a short methodological detour. Because that's a method I will be also using a lot in the future. I've been using a small X-ray machine in Japan, where basically you put there an sample, and it's like taking a chest X-ray, but you take one. But with the insect, we take an X-ray, rotate it, X-ray, rotate it, X-ray, repeat it like 2,000 times, and then the computer combine these images into three-dimensional representation, and we get both external anatomy and internal anatomy in high resolution, typically around 1 micrometer. So the voxel, that's the three-dimensional version of pixel, it's like 1 by 1 by 1. So this method we use to, just scheme what I described you. That's the method we use to then quantify the volume of the muscles. And from the videos, we had access to the speeds and acceleration. We could calculate force thanks to weighting the mandibles and then making some physical model of the mandible, approximating it as a rod, which rotates here around the hinge. And interestingly, when we calculated the ratio of the calculated force to the average closer mandible volume, this seems to converge with an exception, and this might be actually my methodological error. I'm now or analytical error. I'm now looking into this one species. It seems to converge to a certain value. So quite interestingly, despite the termites evolving this strategy probably multiple times independently, they converge at some performance. And we think that might indicate that there's some sort of biological limit. The thing is, they could probably snap even faster, but then your mandibles might just continue after the snap. There is a lot of energy, especially if you miss. If you hit, that's okay, because a lot of the energy is absorbed. But if you miss, the mandibles continue rotating, and then they might just tear out of the head and continue flying. So that's not what you want to have. So this probably imposes a biological limit on how fast you can go. So it might be that the termites like independently reach the biological limit. So I think that's quite a nice example how like looking at something in this phylogenetic framework can show the limits of like some biomechanical adaptation. And I will skip here some more details. And I see I was very ambitious with my talk. So now finally, a lot of the methods I already introduced, that's what I will be doing here, but not with termites. I still will continue working on termites. They are one of the greatest animals in the world, I agree. But there are some which might be even more cool. And these are beetles and flies which live with termites. So here is one example. Yeah, I could have asked, but which one do you think is termite and beetle? But this is termite, this is beetle. This is row beetle from the Allocarina subfamily. So termitophiles many times independently invaded termite societies. Apparently, that's a great lifestyle. Like once you manage to convince termites that you are also termite, or somehow manage to trick them, you have them access to their nest. Termites can feed you. It's defended from predators, from parasites. So pretty good lifestyle. So, and given the large abundance of termites that evolved many times independently, both in beetles. So this is very weird beetle, and this maybe even more weird. This is fly. This is Herbilca, scuttlefly, Foridae. So some very weird morphologies evolved, and by another example of termitophile. But the interesting thing is that, so here, for example, we have these examples of this very fat, this is very fat beetle, very fat beetle, very fat beetle, this is very fat fly. Very similar adaptation evolves again and again and again. The other adaptation of Termitophilos beetle is this, they call it limuloid shape. I think it's because it resembles the horseshoe crab, and its Latin name is something like limu something. So many times convergent in evolution, similar morphologies evolved. So I think, and apparently also the people who gave me money to do this research agree that this is a quite remarkable model to study convergent evolution, because also I look at it as the termite nest being some sort of, let's say, aquariums or terrariums, where, so that means the positions are, the conditions are quite stable and quite comparable across the termite nests. And basically somebody started to run an experiment for us some 100 million years ago. And many times independently, beetles switched from their ancestral, so rove beetles normally you can find them in leaf litter and so on. They are not living symbiosis with termites, but many times independently, and that's all the red branches. And this is only a snapshot. Now we know that it was more than 25 times that beetles switched from the free living to Termitophilos lifestyle. And then often they start to look very similar. And in addition, they are in this environment of termite nests where, and that's my hypothesis, where the proportion of, let's say, stochasticity, that means, for example, how random events influences whether you survive or not, I think is rather low, because it's this protected termite nest. So I think we have very good chance to see that some of these beetles were evolving along pretty much parallel trajectories, and not only morphology-wise, but also here, again, I will skip something. And yeah, here only, that's an example of the result of the microtomography, the outer morphology. So not only that these beetles were evolving in a similar way concerning their phenotypes, but I believe also concerning their genotypes. So there are two possible hypotheses. Either they reach the very similar phenotypes by similar, let's say, for example, same gene families always expanded, or some similar genetic changes always occurred, or different genetic trajectories led to the same morphology. So that's basically what I'm trying to test, and I believe I will find some remarkable parallel genetic trajectories, which I think could adjust quite a bit how we think about predictability of evolution. So I hypothesize the evolution is relatively predictable in these systems, but for that, yes, some more examples of the microtomography data. For that, importantly, and I might then return to the previous slide. Large part of my project is assembling genomes. So this is a graph of completeness of human genome. Maybe you heard that the first announcement of completed genome was here, and all these other points along the timeline, these are just like other versions of a complete genome. So actually really assemble, and you might have heard quite recently, I think it was, what was it, maybe last year, that the really complete human genome was finally sequenced. So apparently sequencing a complete genome is not so easy. That's because there are, for a long time, we had mostly methods which can sequence short stretches, and then that's a problem because large portion of eukaryotic genomes are some repetitive regions, for example, or regions where the repetitive motif is longer than the small piece of DNA you can sequence. So then it's not possible to, in a single unique way to assemble these short reads into some complete genome. But that's now becoming fairly much possible with one method, the PacBio hi-fi sequencing, which produces reads which are long up to 10,000 or up to 20,000 base pairs, and it achieves high accuracy thanks to this neat trick that it circularizes the DNA molecule, and then it reads again and again and again, maybe up to, like, for example, here it's 4 times, and then it creates a consensus. So each time there is some errors, but after you read the same thing like 5 times and you check what's the consensus, you can achieve very high accuracy. So that's a nice method to get high accuracy long reads. And then second method, which I will be using, or I'm already using, is this rather enigmatically named chromosome conformation capture, but very elegant method, where you take an intact cell, you pour some formaldehyde onto it, and so in the cell, in the nucleus, the chromosome is packed in a way that the DNAs which are next to each other are not always also next to each other along the sequence, but they are, if they are close to each other, there is a certain possibility which can be expressed like fully quantitatively, how far from each other along the DNA strand the parts of the DNA are if they touch each other. And with this method, you can basically check which parts of DNA are touching each other. So you use the formaldehyde, you cross-link DNAs which touch each other, then you break it, and you see, but you leave this cross-link where the DNAs were, let's say, touching, and then you sequence it, and you will learn what's the sequence of this portion which was touching this portion. And this will give you some long-range information, for example, saying that this part of DNA, which is hundreds of thousands of base pairs away from this one, is still only like hundreds of thousands, not on different chromosomes or not millions of base pairs apart. And combining this information with assemblies from these reads, we are getting some like near complete genomes comparable to, for example, the latest version of the human genomes. And with these genomes, I will be then doing the comparative genomics. So I skipped a couple of things. I will spend last 2 minutes only explaining. So I introduced you the comparative genomics. That's one method I will be using. Then phylogenetics. I will be doing some large-scale phylogenetic reconstructions using these ultra-conserved elements I introduced in the beginning and up to 2,000 raw beetle specimens. And then I will be also, I started to work on with Thomas van de Kamp from the Karlsruhe Institute of Technology to work on high throughput microtomography of these beetles and also flies. So that's enabled by Thomas having or developing this really nice imaging facility. In Karlsruhe, they have synchrotron, a lot of beamlines. Beamlines exit from the synchrotron, but one of the beamline is dedicated to biological imaging. And they have a robotic arm, which can currently use sample holders with 40 samples and all this. And the sample could be, for example, a Pendorf tube with an insect specimen sitting at the bottom, and it can take the Pendorf tube, put it in front of the exit of X-ray from the synchrotron. And since the X-ray from synchrotron is very energetic, the scan takes 1 minute or 2 minutes. When I was doing these scans in Japan, it took overnight. So with this, you can scan. It's starting to be realistic to really scan like 1,000 specimens, and then try to understand how the phenotype evolves or how it converges. And so that's the third method, comparative genomics, phylogenetic reconstruction, and this high throughput microtomography scanning. And for that, it's then, of course, extremely laborious to work with microtomographic scans because it's it's not like working with images is hard because there is a lot of data, but this is like a three-dimensional image, so there is even more data. I've been doing this so far, let's say sort of manually with a large number, with a small number of samples, but I hope that one of the PhD students I'm recruiting will be able to use some AI-assisted image analysis, and I'm not really able to develop it. There are some tools which were already developed. I understand it just a little bit. So, but we hope we will be able to deploy this to analyze this, these large data sets of the beetle scans, and all this to basically understand in these terrible beetles and flies to what extent they really evolve in a predictable way, and to what extent they have some lineage-specific, unique solutions, trajectories. And with that, thanks for your attention. I'd like to acknowledge Thomas, of the group where I spent my last 6 years. It was really good time. Honza, who introduced me to termites some 8 years ago, and quite some of these things were sort of stimulated by being in the field with him, and him saying, "Oh, look, this is quite cool. I think that might work like this," and without actually anything being published about it, so that stimulated quite some of the research. And there are many other people who provided the samples. I do a little bit of field work, but most of the samples which enable my research were collected by other people, and without them it would not be possible. And thanks for your attention. I think it was pretty long. We will see if we have time for discussion, but from tomorrow I will be here, so feel free to stop by and check anytime. Thanks.
Very interesting system. I wasn't aware of this demo.
Yeah, no, this is very much fun. So there have been somebody who studied them in the 60s or 70s, and now basically not many people are studying them.
Do you know how specialized are these interactions? Are the people specialized in some species of
termites? Yeah, it's pretty much they are pretty much specific. So there can be then that hopefully we will publish soon. That's a manuscript in preparation about the coevolution of beetles with the termites.
Yeah.
So there are then some swaps. So it's not that it would be the phylogenes of the host and the beetles. It's not congruent like across, for example, 100 million years of the evolutionary history of the beetles. So then there are some swaps, but at smaller time scale, it's pretty much congruent. So they stay with the host, and typically one beetle species with one termite species. So it's not like it probably need to be adapted in quite specific way. But then also, but on the other hand, one termite species can actually have quite some heterophile species.
a bit like plants and herbivores?
Sorry?
A bit like plants and herbivores.
Yes.
Specialized herbivore, but one plant can host many herbivores. Yeah.
Can some of these termites derive any benefit from these associations, like a mutualism, or are they just commensals?
Yeah, it's a very good question. We think it's mostly commensalism. There is for sure some parasitism. There might be some mutualism by some of the termitophiles cleaning up like a debris in the nest of termites, but surprisingly, most of the time they have very low abundance in the nest with termitophiles. So you can, that's also not really helping with the research. I guess one of the reasons why people did not study them that much. Like you can dissect and extract a termite nest with tens or hundreds of thousands of individuals, and you can find there like few tens of termitophiles. So especially with these beetles, the abundances are fairly low. So I don't think there is much I mean, organisms within the colony. So I don't think there is probably much, like, let's say, fitness, cost, or benefit to the termites. At least, yeah, I skipped one project where I'm looking at the flies which parasitize inside of the head of termites. So there, and manipulate their behavior and development. So there we know they can actually overtake the colony. So the colony mostly it's just also a couple of individuals, but sometimes basically every second or third individual can be parasitized. In the fly system where the flies are.
So the termite is parasitized.
Yes, by fly, which the larva develops. So that's a bit different system where we know it can really have massive fitness costs, but normally it seems sort of irrelevant, but there is also quite not that much known. So yeah, I'm mostly using it as a model system, and but otherwise, like, I would be very happy if there is some ecological importance for these termitophiles, but I'm skeptical about it. I think it's interesting model for convergent evolution, but yeah.
So also about those termitophiles, I saw in your phylogeny that you also highlighted mimicophilous lineages, so and symbionts. So if within a lineage of one of the termite symbionts, it's also more likely to become mimicophilic in that same lineage.
Yes. So let's jump quickly to it. There are some transitions. Yeah, I completely skipped, and because there are also, I'm trying to navigate around myrmecophily because there are other people like, for example, Joe Parker, more importantly, who is doing great research on myrmecophiles. So I'm trying not to get too much into competition with him. So you can see here really some very probable examples of, for example, here, transition from blue myrmecophily to red termitophily, and perhaps here another from myrmecophily to termitophily. I think we did not see any example of like this switch from termitophily to myrmecophily. But yeah, these switches also happen.
So the next question would be, would they go into similar morphological adaptations if they are within ants and within termites?
So this limuloid, this defended body shape where the pronotum is completely covering the head and so on, retractable appendages, short antenna. That's also present in quite some myrmecophiles. But then there are also quite unique different strategies in myrmecophiles. There is, for example, this but it's actually somehow similar to the mimicking the shape. So some of the some of these fat, and I did not show a real example, but some of these fat rove beetles mimic by their body termites, and it's tactile mimicry. It's quite obvious, nothing is known about it. Everybody was always saying that the shape doesn't matter, it's only pheromones, but yeah, I should have shown some examples, but they are really beetles which have, so this is actually what you see, that's like an abdomen which is, it's bending the abdomen above the head, and it can have on the abdomen 3 pairs of pseudo-appendages, really resembling the shape of the termite. So that's quite obviously to trick the termite, the sensory or the tactile input. And then there was a little bit of research on the chemical ecology, but there is for this tactile mimicry, and in the myrmecophiles, that's why I'm saying it, then there is also a lot of myrmecophilous beetles which very much resemble their host termites. It's most common in the various driver ants, where there are also, I'm not sure if that's resolved, there are hypotheses that because they often migrate with the ants, that's basically, they might be not to trick the ant, but also to trick the predator, because if the predator would see that there is a there is a myrmecophile which is not defended, marching with the ants. It could be, for example, like by a bird picked selectively, but if the myrmecophile is looking very much like an ant, then that's this hypothesis is helping.
I was actually involved in one of those studies from Christoph von Bern, where we looked at the macrophiles and the integration into Aryan colonies, and we found that the most important characteristic that defines like aggression towards the macrophiles is like the body size.
Mhm.
So it's not even like how well they chemically mimic them or something like that, but it's like the shape for sure plays like a really big role.
So if you have the overall dimension is matched to the host.
Yeah, but how it works, we don't really know because the sensory, like the ants cannot see them really well, so not sure exactly how it works that they get so perfect in mimicking or that they have to be so perfect. But
It will be probably a mixture of strategies, I guess.
yeah,
You try to when you come in, and usually their whole of these termitophiles and myrmecophiles, their whole life cycle is not with the host. They have part of their life cycle they disperse, so then they somehow need to get in. So I guess having the proper size and shape helps you maybe to survive just long enough to, like, for example, acquire some particular hydrocarbons through interaction. So it might be like multi-step
yeah,
sneak in a process. So yeah, I remember that paper on the size, that's for sure true. Then, but then also the particular shape that's apparently also playing a role. And then there's chemical mimicry, either acquired from the colony or perhaps also some of it can be de novo synthesized by the beetle, but that seems to be not very probable. So more likely just trying to stay long enough with the host to acquire their particular hydrocarbons.
Are there any other termitophiles except beetles and flies?
Yes, there is a lot of them. So the most diverse group is beetles, specifically Alocarinae, is one of beetle subfamily. Then it flies Florida, but there is a lot of, for example, like millipedes or small centipedes. What else is there? These are the most common. Then it's usually so myrmecophiles are sure crickets. You can see also in Czech Republic a lot of myrmecophilous crickets. I know I have a hard time to remember, but they are quite some other. So I bit simplified by focusing only on, but these are the most diverse. So it's also easiest on them to study to like get really large number of independent evolution and compare across these.
Okay, so thank you. See you around.
Thanks.